Hi there, and welcome to another video by OmnesRes. Today I'll be showing you a very useful tool that we use a lot. So without any further ado, I'll just go ahead and show you what I'm talking about. What I have here is two Python dictionaries. So I've got this dictionary called mRNAs. You can search for any gene name you want. These are human genes. And there's going to be a list of lists. The first entry will be some very useful information. So you've got the name of the gene, you've got the Entres ID, the chromosomal location, and gene synonyms. Now, where this gets really cool is we have a second entry in this list, and if we look through those entries, we can see what we have. Gotta put in there. So here we have every single ref-seq transcript for the gene TP53, also known as gene 53. And we have the accession number of each transcript variant. We have the length, and we have the start of the coding sequence and the end of the coding sequence. And where this gets really useful is when you have a second dictionary with the keys being the accession numbers. And there you have it, the sequence of that transcript. So let's just look at the length of these dictionaries. So I have 20,000 genes, ref-seq genes, and those 20,000 genes have 70,000 transcripts. So we're going to go through how we get these two dictionaries, how you might get different information if you want more information than we have here. And this should be applicable to pretty much any organism that NCBI has in its database. Okay, let's get started. So you just go to NCBI's nucleotide page. Normally you would search for a gene at a time. For example, we just did TP53. And you would click on the organism that you're interested in and the transcript. And you get this block of text. And you can see how we had some of this information in our dictionaries. So we were able to extract all the information we were interested in from data like this. And here you can see the CDS position. And then if you go down far enough, you have the sequence. So that's if you want to look for one gene at a time. But if you know how to use this search box, you can actually get every single RNA. So you can do biomolecule RNA with the properties tag. And in addition, you can choose your organism, and you can use the primary organism tag. Okay, and now that gives us 99,000, almost 100,000 mRNAs, which are ref-seqs, which is larger than the dictionary that I just showed you because they continually update this, and it's been a little while since I downloaded the most recent search. So you can send this to a file. You're not going to want summary, you're going to want GenBank full. And this file is going to be over a gigabyte of data. And if you don't select ref-seq and you choose all RNAs, it's going to be even larger. And if you're looking at DNA, it's going to be huge. So you have to be careful with that. So once you have your file downloaded, the first thing you have to do is open the file and see what we're working with. So we're going to go ahead and do that. Let's run that. Here I have my Python text editor as before, and I have my Python shell on the right. So I just like to see what the file looks like. This is what we're dealing with. Let's make that a little bit bigger. So we have this huge mess of text. And we have to extract the data that we want. So this is going to be a little bit of a longer video, so you might want to fast forward through some portions of it. But we are going to be going over some advanced parsing techniques, and if you want to alter the code, you may have to refer to this video. The parsing code will be available on our website if you just want to go ahead and download it. So how might we go about parsing this file? Well, first thing we need to do is make an empty list to store the data that we're going to be parsing. So we'll do that. We're going to make a variable called x equals f.readline. So we are at the very beginning of the file. And this first line actually has some good information in it. So we're going to take some of that information. But first we're going to make basically an infinite loop by continuing until x is this empty string. And what we're going to say is if locus right here, if it's I know you could use a regular expression for this, but just to make it simple, if it's the very start of the line, do this. So the length of the gene is actually right here, and I do like that information, so I'm going to grab that information. If you don't need that information, you could skip that step there. So we're going to just split. Split just splits on any white space. So this can get split, that's going to get split. And we're going to be taking the 1, 2, 3 third entry, but Python coordinates start with 0, so 0, 1, 2. So we're going to be taking the second entry there. Then we're going to do x equals f.readline. And I'm really interested in this definition. I really want the sort of full definition of the gene. As you'll see, we can get this name later in the file. And then I'm also interested in what transcript variant this, so this is transcript variant 2. I'm very interested in that information as well. So the problem is that, you know, you might say, just go ahead, split on this parentheses right here, but I'm not positive that, you know, there's not going to be a parentheses over here somewhere for a different gene name. I also don't know if there's going to be a parentheses over here or not. So I'm actually going to cheat a little bit and just ignore the definition for now until I have more information to help me split that name. So for now, I'm just going to actually save it in a variable called raw definition. And I'm going to split on definition just to get rid of this. And I'm going to take the Python coordinate 1. So basically this would be 0. Well, it would split on that, so there'd be like an empty, the list would be an empty string, and then it would be the rest of this. And then I'm going to do, and that would only be for that line actually. So I want the whole definition, which can be several lines, and actually if this name's really long, it could be who knows how many lines. So I'm going to have to do something a little bit more complicated. So I'm going to have to basically make another infinite loop that's going to just keep on going until accession is in X, because that's when I know my definition is done. So while accession not in X, take my raw definition, which I instantiated up here, and add to that string. I want to add a space, because you end with this new line and then you just sort of immediately start the next line. So I just want to add a space just to make sure that there's a space between the words, and then I'm going to do plus x dot strip, and then I'm going to say x equals f dot read line. So that's going to continue until it gets to the next line, which is accession. So then once that loop finishes, you know that accession is in x, basically. So then I want that accession number, so I will go ahead and take that accession number right here, and I'll split that. So I just stripped the new line character, I split on the white space that was right here, and then I just took the second entry, Python coordinate 1. Now it gets a little bit interesting because I have a bunch of stuff that I don't want. I just want to like skip all this stuff, but then I have stuff later on that I do want, and yeah, it's a little tricky. So I'm going to use a trick. So I'm going to have this big X equal to false, and then I'm going to say while X equals false, while X is false basically, continue doing a bunch of stuff. So I want a couple things here. So I want the gene name. I definitely want that. So I'm going to go ahead and grab that. So if I see that gene name right there, if I see that, if that's in X, then I'm going to say gene equals x dot strip dot split that gene, the second entry, and then I'm also going to get rid of the quotation marks. Okay, great. So I just grabbed the gene name. And then we have a couple other things that I'm interested in. There's gene ID somewhere in here. See if I can find it. Yeah, there it is. I'm interested in that, and instead of typing it, I have the code already. So I'm just going to show you what that looks like. So if gene ID in X, my entrez ID is that ID. So entrez ID is for the gene. Each gene has one entrez ID. So all the transcripts for this gene will have the same entrez ID, but they will have different accession numbers and different transcript names. Now, the gene synonyms is pretty interesting to me. You know, sometimes you'll be working with a data set, maybe it's a really old data set, and you'll be trying to convert the names to the current name, and these synonyms may help you do that. And this may span multiple lines. So whenever you have something that spans multiple lines, we have to do our trick again. So I'm just going to go ahead and copy that code as well, because it's quite a bit to type. So once we see the gene synonym, we're going to make a big Y equal to false and a small y equal to the line. And we're going to just keep on going and going until we see note. So if we see note, we're going to stop. But you know, I'm not sure that every gene has a note. So I also said, or if we see dbxref, which you can see in here sometimes. So there you go. So that will get you your gene synonyms. And then I'm also interested in the chromosomal location. That can be of interest when doing bioinformatics analyses. So the code for that is there. When you see that in the line, go ahead and grab the chromosomal position. Now we have another interesting thing. I want to know the coding sequence, and I'll go ahead and just paste that into here. Oh, it's too big for my window here. That's how complicated it gets. So I have this if join in X. So where does that come from? So sometimes a couple of these genes, like let me just show you. Okay, I'm going to have to be a little bit more specific. So if CDS, if CDS, NI, and join NI. See if this works out for me. I think I need to reopen the file. Yeah, okay. What happened there was when you are going through a file, it saves your position in the file. So when I looked through the file the first time, I actually already passed the lines I was interested in, for example here, but then I had too much information and I got confused. So then I was more specific and it gave me only the information I needed. So once in a while you have these CDS lines where you have join, and you know, I can't explain why they're there. So I have some code that will deal with that. Most of the time join is not in that line, as you can see from CDS. Normally it just tells you the start and stop. I really don't know where that join comes from, but that code deals with it. I hope you never have like a triple join. I'm not exactly sure if that will deal with triple join or not. So now we want to get the sequence, not the protein sequence. We want the nucleotide sequence, and so that line starts with this. So I have code for that. I'm just going to have to make this full window now. So basically we start with an empty string, and again we don't know how many lines the sequence is going to take. So while X does not equal this interesting character here, this is double backslash, we're going to just keep on adding to the sequence, but we have to remove. So we have these numbers at the beginning that we have to remove, so that's why we've got that 1 to the end, and that takes care of it. And then you'll notice here that we set X equal to true, which means that we're finally going to get out of this loop, because once we get our sequence, that's the end of that transcript entry. We're done with that transcript. So then we finally get out of our loop here, and now we go back to our raw definition of the gene. So we make this variable called gene name, which is, I don't know what to call it, but let's call it definition here. It's not this sort of abbreviation. It's this right here. So I want that, and what I do is I use the fact that I figured out what this was here, gene. I figured out what that was, and then I added some parentheses to it, and now that lets me confidently split my raw definition. I know for a fact that I can split my raw definition now into this name, which I'm interested in, and the transcript variant, which I'm very interested in. So I get the gene name, I get the transcript, and now I'm finally ready to append all this to raw data. So raw data is going to get the gene, which was this, the gene name, which is this thing. Maybe I need a better name for that, gene description. The Entrose ID, the chromosomal location, synonyms, transcript numbers, so which transcript variant, in this case transcript variant 1, the transcript accession, the length of the transcript, the CDS start site, the CDS end site, and the actual sequence. Okay, that was a lot of stuff. I actually need a couple of, let me just make this full screen again. I need a couple x equals f dot read lines, as you might imagine, to just keep these infinite loops going. Okay, and that should run. You'll have to just take my word for it, I guess. I need to move this one more over. There we go. Now, whenever you're parsing, so this is a really large file, and this code is going to take forever to run. Whenever I have code that's going to take a long time to run, I don't want it to have to run again, so you might want to just go ahead and write this parsed to text. So what I like to do is I like to just go ahead and make a file, and then for I in raw data, and then for j in I can just write each entry, tab delimit the data so that it's easy to read later, and then have a new line after each line of data, and then close the file. So I will go ahead and run this just to make sure it's working, but I don't want to let it go all the way. Nope, we did something bad here. Oh, I don't know why I have a splitting on an empty character there. Okay, looks like I actually typed things correctly now. So I don't want that to finish, so we're just going to take a look and see what the first couple entries of the list looks like. So what do we got? So let's see. So yeah, this is the first entry. We have the gene name, we have the description, so everything is looking great. So how do we get from this parsed data to our dictionaries, if you want to use dictionaries? If you're happy with lists, then I guess you're done. But dictionaries are a very fast way to access information. So what I did is I just created two dictionaries, mRNAs and sequence. You might want to title this sequences or whatever you want to call it. Raw data. So I have to read that file I just wrote, or was in the process of writing, if I let the code finish. And I'm going to append this list comprehension here. And then for I in, so basically I just created all of this. I just recapitulated this basically. This is what I have again. I had to go to -1 because I had like an extra tab at the end when I wrote the file. So for I in raw data, if mRNAs .has key, so basically I want to accumulate all the transcripts for each gene. So if you have the key, if that is false, if you don't have the key yet, do this, grab this information from the raw data. I'm missing one bracket. And then go ahead and append the rest of the data. So basically I'm creating an initial entry, and then I'm going to append to that another list to make a second entry in the list. And I don't want the sequence in my mRNA's dictionary. I want the sequence in the sequence dictionary. There you go. And if that mRNA already has a key, then do this over here. Append to that second entry, that second list. Okay, and there you have it. This should run perfectly. The code on the website for download will be absolutely correct. I'm not sure if I typed everything perfectly here. But thanks again for watching, and if you have any problems you would like us to tackle, please let us know in the comments below.